All right, so we're going to take a quick look at 7.5. You should be familiar with the information in the rest of Chapter 7 just from previous courses. It's basically going through biological molecules, so talking about carbohydrates, the structure of carbohydrates, how they're built, how they break apart, talking about lipids, the structure of lipids, how they are a part of our cells and bacterial cells, prokaryotic and eukaryotic organism cells. Also looking at proteins and how they're structured, the amino acids, and then how those build chains to then fold on top of themselves multiple times and then make a fully functional protein. So those are all things that you should already be familiar with from previous chapters, but if you need to, you should brush up on that information in either Chapter 7 here or in other texts from your previous courses. So here what we're going to do is we're going to take a look at how those biological molecules, the carbohydrates, the lipids, the proteins, can be used to identify microorganisms. And we're actually going to look at this in a future chapter as well in a little bit more detail in a way that is not, it's in a little more simple of a way, using different agar plates and different deeps in tubes and things like that. But for here in 7.5, I just wanted to take a brief look at some of the more industrial type ways that these things are looked at. So let's get started. Accurate identification of bacterial isolates is essential. Of course, this is important because for clinical microbiology, we need to know what is in a patient. So when a patient presents with some symptoms and they go to a doctor, then they're going to try to figure out what it is that this patient has. So they'll take some samples depending on the type of problem and the type of symptom. If it's a rash on the skin, they may take some samples from the rash there. They may take some blood samples, some saliva samples, depending on where the organism is and what kind of symptom it presents. So, but it's important that, of course, these things are taken a very close look at and that it is identified very specifically, because that's going to determine what treatment is going to be given to the patient. We don't want to give patients treatments that are not for the organism that they have in their body. So, for example, cases of food poisoning require accurate identification of the causative agent so that physicians can prescribe appropriate treatment. So if you remember back to our Chapter 4 and Chapter 5, there were lots of microorganisms that caused what we would call food poisoning. So many that when somebody presents with those types of symptoms, it would be important for the physician to go in and make sure that they get the samples required to then send to the laboratory so they can determine the specific causative agent so that they can then give appropriate treatment for that. Likewise, it is important to accurately identify the causative pathogen during an outbreak of disease, so that the appropriate strategies can be employed to contain the epidemic. So if there is an epidemic somewhere, it is very important in order to figure out which specific pathogen it is so that it can be deterred. So there are many ways to detect, characterize, and identify microorganisms. Some of them rely on phenotypic biochemical characteristics. So phenotypic is what it looks like or what it presents with, not the actual genetic information, but what the genetic information codes for. While others do use that genetic information, so genotypic identification. So looking at either what it's presented with, phenotypic, or genotypic, which are the actual genes. So the biochemical characteristics of a bacterium provide many traits that are useful for classification and identification. So we can analyze the nutritional and metabolic capabilities of the bacterium, or the bacterial isolate, and that's common to determine the genus and the species of the bacterium. So we will look at this microbial metabolism. We're going to get to that section. We'll discuss it in more detail. But in this section, we're just, as I mentioned, going to discuss a few methods that use biochemical characteristics to identify some microorganisms. So some microorganisms store certain compounds as granules. So remember, we do have organisms that have granules, little pockets of chemicals and different enzymes and things inside of their cytoplasm. So these granules and the contents of these granules can be used for identification purposes. So we know that there are certain bacteria, for example, that have these granules and others that do not. So those granules themselves can be used to identify. So for example, polybeta hydroxybutyrate, which is PHB, is a carbon and energy storage compound found in some non-fluorescent bacteria of Pseudomonas, the genus Pseudomonas. So there are different species within this genus, and they can be classified whether or not they have PHB or whether or not they have fluorescent pigments. So the human pathogen, P. aeruginosa, and the plant pathogen, P. syringae, are two examples of fluorescent Pseudomonas species. So they do not accumulate this PHB in their granules. They don't have any PHB granules, but they are fluorescent Pseudomonas. So actually there's an image of this up here. You can see this is a scanning electron micrograph of Pseudomonas aeruginosa, but here what I was more talking about is that it is pigment producing. So you can see that this P. aeruginosa was placed on cetramide, or agar, and then it shows a green pigment called pyocyanin. And so they know that this is P. aeruginosa because of this green pigment on this specific type of plate. So there are many different ways that we can identify the bacteria or the pathogen in these ways. So other systems rely on biochemical characteristics to identify microorganisms by their biochemical reactions. So the reactions that they're having within this organism, the biochemical, so biological, meaning living, and then chemical, so these chemical biological reactions that are happening inside of the organism, such as carbon utilization and other metabolic tests. So it mentions here that in small laboratory settings or in teaching laboratories like at a college or university, those assays are carried out using limited number of test tubes. So similar to laboratories at SC4, in other colleges and universities, we would use test tubes to run these samples and place things in agar deeps and on plates and things like that. However, more modern systems, such as this Biolog Inc. Company, they make these huge machines that can go through this process in much greater number and much faster than using individual test tubes. So they're based on panels of biochemical reactions, all performed simultaneously and then analyzed by specific software. So their Biolog system identifies cells based on their ability to metabolize certain biochemicals, so their ability to metabolize them, break them down, and utilize these biochemicals, and on their physiological properties like pH and chemical sensitivity. So it uses all the major classes of biochemicals in its analysis, and then it can be performed manually or with the semi or fully automated instruments. So it can be manually if needed, or you can just send this right into the machine and it'll go through all of these different tests in order to determine what organism you have. So one of the machines, or one of these automated systems that identifies these microorganisms, is one that determines the specimen's mass spectrum. So if you've taken some chemistry courses, for example, perhaps you've talked about mass spectrum. We won't go into it in detail here. I just wanted to briefly kind of scratch the surface of some of these things just so you understand that small laboratories, again, may use test tubes and plates and things like that, but a lot of the really large highly processed companies are going to be using things like this. And so you just understand that they're using things like mass spectrometry versus test tubes and a person actually handling the organism. In this case, they use this mass spectrometry and then compare it to a database that contains the mass spectra for thousands of microorganisms. So they use this matrix-assisted laser desorption ionization time-of-flight mass spectrometry. I'm not expecting you to remember all of that, just this MALDI-TOF, basically, and uses disposable MALDI plates. So this right here is more of what you should understand in this process. So they grow these microorganisms on a MALDI plate, and you can see it's like having little tiny wells in there where the organisms will grow. Then what they do, so they add a matrix reagent to it. So they grow the microorganism, they add this matrix reagent to it, and then this mixture is irradiated. So we irradiate the sample. So with this high-intensity pulsed ultraviolet laser. So we laser all of these samples, and then what happens is when they apply this laser, when they use this high-intensity UV laser, it's going to eject gaseous ions. So there will be various biochemical reactions occurring in every one of these little tiny well areas. And then when they use this irradiation, then depending on the chemical composition here, what is in the wells, depending on the reactions that happened, they're going to get different types of gaseous ions that are going to come off of each one of these little areas here. And so then these different chemical constituents of the microorganism are measured using mass spectrometer. So these gaseous ions are collected, and then they are accelerated through the mass spectrometer. So the ions are going to travel at a velocity determined based on their mass to charge ratio. So again, not something that you need to know, basically just their charge, so whether or not they're positive or negatively charged, and their different mass sizes of these gases are going to be forced through this machine, through this mass spectrometry machine, and then it's going to give them a printout of the things that are going through and at what time and at what quantity. So then they can compare what came out based on a patient sample compared to the database, for example, and then they'll know exactly what it is. So this signal that comes through can be compared to these thousands that are in the library. So another way that we can identify microbes is based on their lipid profiles, and that's why it's a part of this chapter where it's talking about carbohydrates and lipids and proteins. So we can identify them based on their lipids that they have. So it says, as we have learned, because previous in this chapter it talked about fatty acids of lipids. So remember, fatty acids vary in chain length, so we can have very, very long fatty acids or shorter fatty acids. Also, the presence or absence of double bonds, so we can have saturated fatty acids or unsaturated fatty acids, depending on whether or not they're single bonds and saturated fatty acids, or double bonds, which would be an unsaturated fatty acid, and then depending on the number of double bonds in these fatty acids, as well as the addition of hydroxyl groups, branches to the fatty acid, and rings to it, and also things like cholesterol has rings and sterols have rings. So to identify a microbe by its lipid composition, it's very similar to what we just talked about. So the fatty acids that are present in their membranes are analyzed. The common biochemical analysis used for this is called fatty acid methyl ester analysis, or FAME. So fatty acid based on the fatty acids of the lipids, and then methyl ester analysis. And that's because they take these fatty acids from the membranes of the microorganisms, and then they chemically alter them to form methyl esters. So fatty acid, methyl esters, FAME analysis. So they change them to form these methyl esters, and then they analyze them through gas chromatography. Gas chromatography then will result in this chromatogram, and then they compare it to reference chromatograms, similar to mass spectrometry, where they get this feed from it. This machine will give them the information, and then they can compare it to those that are already on file that are known organisms. So here's the illustration for that. We have our bacterial cultures grown on a plate, just like we would do in a laboratory. And then they extract the fatty acids and then convert them to the methyl esters. Then they use a gas chromatograph here to analyze this methyl ester fingerprint. So the amount of fatty acids, the types of fatty acids, the length, again, if there are hydroxyl groups or rings, all of those things will be picked up by this gas chromatograph. And then it gives this readout, and then you can identify the bacteria based on this readout, comparing it to known bacterium. So similar to that, a related method is phospholipid derived fatty acid analysis, so PLFA, analysis. So remember that our membranes are mostly composed of these phospholipids, phospholipid membrane. Then what they can do is they can saponify it. So saponify is to kind of turn it into a soap, break things apart. So hydrolyze with alkali. So in this situation, what happens is that it then releases the fatty acids. So we take the phospholipids, we release the fatty acid, and then this resulting fatty acid mixture is then subjected to this FAME analysis. So going back with the gas chromatograph, gives a printout, and then we can identify the unknown organism. Another way to identify is based on proteins. So we just talked about a couple ways to do it based on lipids. We can also do it based on proteins produced under specific growth conditions within the human body. So these are called proteomic analysis. Proteo meaning protein, proteomic analysis. To perform proteomic analysis, proteins from the pathogen are separated by high-pressure liquid chromatography. If you remember, at some point, you've probably done where you've taken the chlorophyll from plants or smashed up some leaves or some grass or something like that, and then you've placed it in a container, and then you put a piece of paper, it may or may not have been a specific type of paper, a chromatography paper, and then you put it in there and you allow the colors to come up through the paper, and it separates out into its different components. This is a high-pressure version of that, of liquid chromatography, where we're separating out the components based on this mixture, based on the size of the components and based on the charges of the components. But the proteomic analysis is going to use this to separate out the proteins, and then they will collect the fractions, which are the pieces of proteins in solution. Then they digest them to make smaller peptide fragments. Then these smaller fragments are identified by mass spectrometry and then compared with known microorganisms. So in a very similar way to the other ones, it's just that then we're using the proteins versus using the lipids. And then lastly, microorganisms can be identified by the carbohydrates. So lipids, proteins, and carbohydrates that are attached to our glycoproteins. So attached to the proteins, those are glycoproteins in the plasma membrane or the cell wall. So antibodies and other carbohydrate-binding proteins can attach to these specific carbohydrates on the cell surfaces and cause the cells to clump together. We can do serological tests, for example, those used for Streptococcus species, to determine or detect the unique carbohydrates that are located on the surface of the cell. And we kind of already discussed this in the chapter on viruses, but it was related to the spikes or the glycoproteins on the virus's capsid. And in that case, we used the red blood cells or erythrocytes, and we either saw or didn't see a clumping, an agglutination. And that was a serological test, and we can see that done with bacteria as well and other microbes or pathogens. So one way to do that is by separating these carbohydrates out and then using antibodies. All right, so again, that's just kind of a general overview of some of the more boisterous or the larger ways that people or companies oftentimes go through and can identify microorganisms based on the different type of organic molecules that they contain. And we'll look at a lot more of these identification type things in all of the chapters that we're going to be going through forward.